Hello, I'm Robin Brumble, and I am Director of Scientific Affairs and Research here at CLL Society. We are entering into the third year since the start of our research program, for which in just 3 short years, we have already committed to funding $850,000 solely towards CLL and SLL research. Today, I am joined by our 2024 Clinical Scholar Award recipient, Dr. Jennifer Brown. And this particular award is the largest in size thus far for CLL Society, and we are extremely thankful to the many individual donors in our community who have helped to make this award possible. Our Clinical Scholar Award is dedicated to an exceptional physician researcher who has already established themselves in the field of CLL and SLL, and it's designed to support the research efforts of an advanced physician scientist who is engaged in independent clinical research and has demonstrated exceptional leadership, scholarship, and academic success. The overall goal of this particular award is intended to advance the development of treatment options for those living with CLL and SLL. So, Dr. Brown, at this time, I would like to first start off by saying congratulations. And if you could maybe begin by telling us a little about yourself, and I would love to hear about what led you to the decision of dedicating your life's work to helping individuals with CLL and SLL.
Sure, thank you. And I'm very excited to have received this award from the CLL Society. So I am currently director of the CLL Center and Institute Physician at Dana-Farber Cancer Institute, as well as the Worthington and Margaret Collette Professor of Medicine at Harvard Medical School. I grew up on Long Island outside New York and went to Yale and then came to Harvard to do my MD PhD and have been here more or less ever since. I initially fell in love with hematology research in medical school. I really enjoyed looking at the cells under the microscope. And as I trained more in medicine, I discovered that I particularly liked studying blood cancer a little bit more than benign blood diseases, and was particularly interested in lymphomas because of the sort of wide range of disease behavior, and recognizing, of course, that CLL biologically is a lymphoma. It's part of the same spectrum of disease of lymphomas. And I had decided to focus on what we call the lower grade ones, which include CLL. But then the opportunity arose to focus solely on CLL. And I was particularly interested in that for a couple of reasons. One is that I was interested in trying to study the cells in the lab and understand more about them. And that's really easy to do with CLL because of the cells circulating in the blood. Most people are happy to donate some extra blood for research. It's much easier than with lymphomas that are based in lymph nodes, where the biopsies are much more invasive. So it's much harder to get tissue to study. And I've always been interested also in the inheritance of lymphoma. And CLL, it turns out, has a higher heritability than other lymphomas. And so that fit in very well with the work that I was starting in looking into the inheritance of CLL. And it also turns out, you know, it's a very variable disease, and there are a lot of manifestations that are somewhat unique compared to some of the other blood cancers, and that we still don't understand. And so that means that there is a lot of work to be done and a lot to be learned, and that interested me, obviously.
Thank you. Well, your research project is going to look at combining the drug Roginolisib in combination with Venetoclax and Obinutuzumab for those with CLL who have had disease progression after being on a BTK inhibitor medication. And for those in our audience who are watching today, BTK inhibitors include medications such as ibrutinib, acalabrutinib, zanubrutinib. But first, let's talk a little bit about Roginolisib. Maybe if you could briefly touch upon what kind of medication it is, how it works, and what we know up to this point in time about its use.
So it's a targeted inhibitor, and it's a targeted inhibitor that blocks the PI3 kinase target. As people may know, there are a couple of approved inhibitors against PI3 kinases, but they've been troubled by a lot of toxicity that is a little bit unpredictable, that's made it hard to manage, and so they're not as widely used in CLL at this point. Roginolisib is novel because it's a new class of drug. Even though it inhibits the same target as that older class of drugs, it's a new class in terms of its chemistry. It works in a different way. Instead of sort of directly inhibiting the protein target, the PI3 kinase, it kind of binds to it and changes the shape of it. And that has different downstream effects. And so it's the first example of that type of inhibitor that has made it to the clinic to inhibit PI3 kinase. And so we're pretty excited about being able to test that in CLL.
Well, you did touch briefly on some of the other PI3K inhibitors that have been used, and there have been concerns, particularly surrounding the drug toxicities that can occur with their use. But do you anticipate some of those same drug toxicities occurring with Roginolisib at all, or how might this medication differ in terms of side effects and toxicities from other PI3K inhibitors that have been used?
Right. Well, we're certainly hoping that it won't have those toxicities. And there's a reasonably good rationale for hoping that it won't. So one is what I was just saying about it being a very different chemistry. Another factor is that in the body, it really just stays as itself. Many of the other drugs get turned into other forms that can sometimes have unexpected side effects. And so this one has a very clean, we call it metabolism in the body. So that's another thing that's in its favor. And then the data that we do have from the initial trial that was done in Europe, it was mostly in solid tumor patients, but there were some lymphoma patients. There was really very limited toxicity and none of the kind of autoimmune toxicity that we saw with the earlier generation of PI3 kinase inhibitors. So that's why we're hopeful that as we move into CLL patients, we will continue to have that same lack of toxicity. And I think there's every reason to think so, because the older drugs, even in the types of patients who were studied in the first study with Roginolisib, did have autoimmune toxicity. It's not that that was something specific to CLL with those drugs. So hopefully we won't have that problem.
That would be very exciting. So I know you've done a lot of research in your career, but what led you exactly to wanting to research this particular drug combination at this point in your career?
Mm-hmm. Well, I've had a long-standing interest in the PI3 kinase inhibitors. I was involved in the initial phase 1 study of Idelalisib when we were witness to its remarkable ability to shrink lymph nodes under our eyes. and then as time went on, the toxicities became evident and trying to manage. I've actually been studying a lot the mechanisms of the toxicities from some of our earlier clinical trials. And so I was interested in pursuing Roginolisib because of the opportunity to target the same target, which is a very important target for CLL patients, I think, that we're not adequately making use of with the existing drugs because of the toxicity problems. And so hopefully, as we move to Roginolisib, we can start to better utilize the effectiveness of this target for treating CLL. And we've been studying it in the lab. We've been studying it together with Venetoclax in particular. And we see that the two of them work together very well, much better than either alone, something we call synergy. And this is consistent also with a lot of pre-existing data combining PI3 kinase inhibitors with Venetoclax in other lymphoma type model systems. So we know that this is a very encouraging combination. And we've also actually done a study at Dana-Farber, which was led by my colleague, Dr. Matt Davids, with one of the earlier generation PI3 kinase inhibitors, Duvelisib, in combination with Venetoclax. And we had very good results of that study. We had very deep remissions, even in people who'd had prior BTK inhibitors, very high rates of what we call undetectable minimal residual disease, where we can't find the CLL cells anymore with our existing technologies. And so there's a lot of rationale for studying Roginolisib with Venetoclax. And then adding the antibody just makes a lot of sense because we know that it's very effective and it will be useful, particularly to get data with Obinutuzumab in people with CLL that's relapsed because it's mostly been used frontline. But we think it can benefit people who've had prior treatment as well.
Great. Well, I think some people might be interested into the logistics of the research project itself. So maybe just touch briefly about what it is exactly that you're going to be doing with maybe like the methodologies you will be using in the way that you have this research set up.
Right, well, so since the earliest trial had already been done in Europe and there was really very little toxicity, I was hoping to be able to evaluate both the safety and the effectiveness of Roginolisib in CLL patients in a way that would allow us to potentially even move it toward FDA registration in CLL. And so that, together with the fact that we know that there's this very effective, what we call synergy in the lab between Roginolisib and Venetoclax, I wanted to combine Roginolisib with Venetoclax and Obinutuzumab. But then the way we're doing that, we're actually going to do something called a randomized phase 2 trial. And so that means some people will get the Roginolisib with the Venetoclax and Obinutuzumab. Other people will get just Venetoclax and Obinutuzumab. And so that way we can assess exactly how much extra benefit the Roginolisib is providing. And we think that will be significant. And then if it is significant, it provides actually proof of principle to potentially do a phase 3 trial that could serve as a registration mechanism. So that's why we're trying to design the trial in this way.
Great. Well, do you have any other final thoughts that you would like to share as we close?
Well, I'm very honored to have received this award from the CLL Society. And I think it'll be very important, hopefully, in allowing us again to regain this targeted class of inhibitors against PI3 kinases with Roginolisib. So thank you.
Well, Dr. Brown, on behalf of CLL Society, I would like to thank you for all of the work that you're doing to advance research in CLL and SLL. And I know that we're all eager to find out about your findings. And I think I can speak on behalf of both CLL Society and the community we serve in saying that we are all very much looking forward to hearing more in the future about your results. So thank you so much for your time today.
Thank you.